Dear friends, welcome to Biology Made Easy. Today we are going to discuss DNA replication. Previously, in other videos, I have told you that the DNA has two important functions. One is autocatalytic, the other is heterocatalytic. In autocatalytic function, the DNA produces its own replica, means own type. That is known as DNA replication. Now the question comes to our mind. What is the necessity of replication? That is very important. Now I will give you one example. Suppose I have 1000 rupees. I want to distribute among two of you. So naturally I will give 500. But I want that you two should have also 1000. How it is possible? In that case, I have to double the 1000 with me, then I can give 1000 each. Again, if you want to distribute among your friends and again 1000, so what do you have to do? Again, you have to double that 1000 to 2000 and again you will distribute. So everybody will have 1000. The same thing happens in case of a cell. We know that when one egg and one male gamete, they combine together, one zygote is formed. This is the starting point of all multicellular organisms. Now, this zygote will divide into two cells and all the cells are equal. The amount of chromosomes, the amount of DNA are equal. How it will be possible? Suppose let us take the example of human beings. In human beings, there are 2n number of chromosomes. Now, each cell will have 2n number of chromosomes and the same amount of DNA. Again, when will they divide into 4 cells, again, in each cell, there will be 46 chromosomes. How it is possible? Unless the DNA replicates, duplicates. That is the aim behind a DNA replication, right? So, when it will replicate, at what stage it will replicate? When the cell is dividing. So, when the cell is dividing, the first is cell cycle. In the cell cycle, there is G1, S, G2, all these phases are there. And this DNA replicates during S phase of the cell cycle. Important from examination point of view. Now, if at S phase it will replicate, what is the method of replication? Again, in the previous videos, I have discussed with you that the idea of replication was conceptualized by Watson and Crick, and experimentally it was proved by Messelson and Stahl. So again, repeating that those things not necessary. Now we have to see how this replication takes place. So for a replication, the first thing, DNA is a polymer of nucleotides. And what type of nucleotides? Deoxynucleotides. And what are the different types of deoxynucleotides? One is deoxyadenosine monophosphate, deoxyguanosine monophosphate, deoxycytidine monophosphate, and deoxythymine monophosphate. These are the different substrates. Now, in the first step, these nucleotides will be phosphorylated with the help of enzyme phosphorylase, and they will be converted to their triphosphates. Deoxyadenosine triphosphate, deoxyguanosine triphosphate, deoxycytidine triphosphate, and deoxythymine triphosphate. Now one question may come to your mind. We want only these nucleotides, but why they are converted to their triphosphate? What is the answer? When these triphosphates of deoxyribonucleotides will be used, they will serve a dual purpose. Number one, they will supply the substrates, and when these will break, the pyrophosphate will be released and it provides energy, because replication requires a lot of energy. That is why these triphosphates they serve dual purpose. This is the first step. Now the deoxyribonucleotides are ready for polymerization. Now come to the second step. Now in the second step, when replication takes place, whether it is eukaryote or prokaryote, always the replication starts from a particular point. That point is known as point of origin. Or in short, we can write ORI, point of origin. In case of prokaryotes, there is a single point of origin, while in case of eukaryotes, there are several points of origin. Why? Because in case of prokaryotes, the DNA is very small size, having less number of genes. But in case of eukaryotes, the DNA is very, very long. Suppose you take the case of chromosome 1, there are 2,968 genes. How long it is? That is why it has several points of origin, so that at several points simultaneously it can be replicated. That is it. Now you come to the third point. Now how this point of origin will be recognized? So that is done by another enzyme, that is a rep protein. This rep protein is also known as DNA A protein. This rep protein or DNA A protein will recognize where this point of origin lies in the DNA. Now you see, I am drawing the diagram of DNA. This is DNA. Now where the point of origin lies? Now this rep protein will recognize that particular point where it lies, and there it will form a bubble-like structure like this. A bubble-like structure. It indicates that the point of origin lies here. Then the fourth step. In the fourth step, what will happen once this point of recognition or point of origin is identified? Now how the replication will start? Now suppose this is 3 prime, this is 5 prime, and this side this is 5 prime, this is 3 prime. So from this point of origin, since this is 3 prime, this will be 5 prime, this is 5 prime, so this will be 3 prime. Similarly, this is 5 prime, this will be 3 prime, and this is 3 prime, this will be 5 prime. From this point, how the replication is taking place? You see, this is 3 prime, and the replication will start from this point and it will continue like this continuously. While on the other side, it will be synthesized in the opposite direction like this, which we'll discuss. In this side, this is 3 prime, so the replication will start in this way, and this side it will take place like this. That means the DNA replication is always bidirectional. It is from this point of origin, it can take place to right side, it can take place to left side. That is important point. Now you come to step number 5. In the next step, what will happen? Now, from this point of origin, the 2 strands of the DNA are to be separated, because unless they are separated, replication cannot take place, because each strand of the DNA will act as a template and one new will be synthesized. So the 2 strands are again connected by means of hydrogen bonds, A and T, 2 bonds, G and C, 3 bonds. So who will break it? That will be broken by one enzyme that is known as helicase. Otherwise it is known as unwindase. and it is also known as DNA A B protein. Now this helicase enzyme, it will break the hydrogen bonds between the nucleotides of the two strands by means of ATP hydrolysis. That is, for breaking the hydrogen bonds, it again requires energy. So now they will be separated, and always it will start this from the point of bubble, which is usually AT rich, because AT in between AT there are two bonds, while in between GC there are three bonds. So it is very easy to break two bonds between A and P. That is why the point of origin you will find there are more AT bonds. So now, when there will be separation, now let us discuss only one side, so that there will be no confusion. Now you are confirmed that it is bidirectional, so we will discuss in one side for better understanding. Let us take this right side. Now you come to step number 6. Now from this point, the separation or breaking of hydrogen bonds between the two strands have started, and it will proceed in this way. Sorry. So when they will proceed, what will happen? These two strands will be separated, and ultimately it will give rise a Y-shaped structure like this. This is 3 prime, this is 5 prime, this is 5 prime, this is 3 prime. Now one Y-shaped fork is formed. Y-shaped fork is formed. So in which stage this Y-shaped fork is formed? During S phase, because the replication taking place only in S phase. So when this Y-shaped fork is formed, what will happen to the DNA molecule? Now, in the DNA, there are two strands, and they are coiled in the right-hand manner. Now, when the two strands will separate, what will happen in the front? They will be supercoiled. All of you have seen the electric wires. In electric wires, two colors, they are winded around each other. If you try to open them, you will find in the front they are supercoiled, and even if you pull them, they will not be separated. The same thing happens in case of DNA replication. When these two strands will be separated from a Y-shaped fork, in the front they will be supercoiled, and there will be further uncoiling is not possible. So then the next step comes, that is 7th step. In this case, there is another enzyme known as topoisomerase. Now this topoisomerase will break one of the strands. It will break one of the strands, because from this point there is supercoiling. So it will break one of the strands, then it will rewind it. Suppose they are coiled in a clockwise manner, then it will be in anti-clockwise manner, it will be rewinded. So now they are free. Again, it will reseal it. That means the same enzyme has the capacity of nicking. Nicking means cutting. And again, capacity of stitching or resealing. Resealing. That means the same enzyme cuts and again joins. Nicking and resealing or stitching. In other words, you can tell that this topoisomerase enzyme has nuclease activity, cutting, and ligase activity, joining. This is very important from examination point of view. So now the supercoiling which lies ahead of this Y-shaped fork, now it is free, because topoisomerase has cut it, rewinded it, and again resealed it. Now it is free, and now replication can continue further. But there is another point, point number 8. In case of plants, this topoisomerase enzyme is not there. There the enzyme is DNA gyrase. This DNA gyrase performs two functions. It carries out the function of helicase and also the function of a topoisomerase. In case of plants and also in case of prokaryotes. That means in those cases, this DNA gyrase will separate the two strands, that is the function of helicase, and again it will cut and resell one of the strand, function of topoisomerase. So it performs two functions in case of prokaryotes. So this again relaxes the supercoiling. Just like that is why sometimes this DNA gyrase is also known as topoisomerase 2. So whether prokaryotes or eukaryotes, now these strands are free and it can continue. But now you come to the next point or next step. Now what will happen? Now when the two strands of the DNA are separated from a Y-shaped fork, always they are coiled around each other. But when we will separate them, how long they will remain like this? There is a possibility that again they will be recoiled. Again we will separate, again they will recoil. Then how this replication will take place? That is why here some single strand binding proteins known as SSB proteins. SSB proteins, single strand binding proteins, or known as helix stabilizing protein, or you can call it swiveling protein. Swiveling protein. So this is single strand binding protein. It is attached to these two strands were separated forming a Y-shaped fork. Now these two strands cannot join with each other. They will remain till the DNA replication is complete. So today we will discuss up to this, because DNA replication is very long, and from this point we will start in the next video. But here whatever I have discussed, I will give you few questions. Now question number 1. Y-shaped fork is found in which stage? Y-shaped fork is found in which stage? This is important from all examination point of view. Y-shaped fork is found in which stage of cell cycle? G1, S, G2, M phase. Second point, which enzyme breaks hydrogen bonds between two strands of DNA? A choice, helicase. B choice, DNA A B protein. C choice, unwindase. D choice, all of them. Now you come to the third question. Eukaryotic DNA replication differs from prokaryotes in having dash points of origin. A choice 1, B choice 2, C choice several, D choice none. I hope you will be able to answer all these questions. Thank you very much. Again, we will meet in the part 2, DNA replication part 2. Thank you very much.